Hello, everyone. Thank you so much for joining us this evening. My name is Jamie Bell, and I'm the Development Director here for JDRF in Northern Ohio. I have the pleasure of introducing Elizabeth Caswell to you all this evening. Before I do that, I just wanted to share that we had a bit of miscommunication with our speakers. Not a big deal. They're coming right from clinic. They're headed our way now. But now we have the pleasure of getting to hear from Elizabeth, who is our International Board of Directors chair for the research committee. Elizabeth is helping JDRF make decisions on how we get drugs and research from bench to bedside. She's here to share all about the JDRF research pipeline. And Elizabeth is from Ann Arbor, Michigan.
No booing allowed.
Oh,
I can go to Michigan.
We didn't plant her to say that. Elizabeth joined the JDRF in Michigan, Northern Ohio chapter board in 2014, and then she joined the executive committee in 2016. She chaired the 2018 and 2019 JDRF Journey for T1D event in Ann Arbor, and in the first 2 years, the event has raised over $300,000. The event continues to be held annually. She's always contributing to our fundraising efforts. She's serving in her 4th term on the JDRF International Board. Her 7th term on the JDRF Research Committee is in her first, and she's in her first year as the chair of that committee. She's also on the JDRF Funding Committee. She enjoys providing updates on JDRF mission and research across the country. And in 2019, Elizabeth received the JDRF Huntsman Community Engagement Award for her service to the organization. So if you do not mind, please help me in welcoming Elizabeth.
So thanks so much for rolling with us tonight. So I thought I'd start off just by telling you a little bit about why I'm here. So in 1991, oh my gosh, that strained my brain to figure out what year that was. I met the first person I knew with type 1 diabetes. He was my next door neighbor when I was in graduate school, and he was a senior at the University of Texas at Austin. And you're never supposed to date your neighbor. Apparently, I broke the rules, but we did start dating, and he really pretty much managed his own T1D. I remember things about that time in his life, like blood glucose testing machines that were about yay big. I sometimes say that about the size of 2 VCR VCR tapes, and like some people just look at me like, what's a VCR? But any of you guys remember those really big blood glucose testing? Yeah, I see some hands. And then he had to roll one of his insulin vials in his hands to make it cloudy. That R and NPH insulin that he used. Anybody remember R and yeah, okay. So that's when I met him. And over the course of dating, we really had some very serious conversations about whether, if we proceeded with our relationship, we might have children together. And I managed to convince him that for me, that was something that was pretty important. And we got married, and we now have 3 boys. My boys are 22, 19, and 17. It's a pretty busy time in our lives, as we have a senior in high school right now. When our middle son was 3, I had probably the single worst day of my life when I had to call my husband and tell him that he was diagnosed with type 1. And when our older son was diagnosed at the age of 12, we were pretty much professionals then, but it didn't mitigate the heartbreak that we experienced. So we got involved with JDRF almost right away when our 3-year-old middle son, who's now 19, was diagnosed, and haven't looked back. And so that's why I'm here. I have a real passion for the vision and mission of this organization. And I thought what I'd do, since we have a little bit of extra time before our panelists get here, is tell you a little bit about the JDRF research strategy, because I am currently the JDRF Research Committee Chair. So I'm your volunteer representative to the organization's research strategy and research department. And it is my honor to serve in that role. I feel very responsible for our community's investment in JDRF and ensuring that we invest wisely with the resources that many of you fundraise, donate, or your time that you invest in the organization. So let me tell you a little bit about how we approach our research strategy. We really are a bench to bedside organization. So what does that mean? Some people had the opportunity to see a little bit of a wet lab upstairs, doing some very basic science, trying to understand things about what causes type 1 or what's going on in the immune system when type 1 occurs, answering some of these very deep quintessential questions that we need to know the answers of so that we can then translate that information from, say, maybe an animal model into a human being. So we go from basic science to or discovery research to translational research, which leads to clinical trials. Right now, JDRF is funding between 70 and 80 clinical trials, and we're funding research in over 23 countries around the world. We go where the very best and brightest researchers are in the country. And right now, there is about $1.6 million worth of research grants right here in Ohio, including the grant that I'm going to butcher Parham's last name, so I'm just going to say Dr. Parham's lab has in curative therapies. A part of getting any drug into the hands of our loved ones ourselves is getting regulatory approval. In the United States, that's with the FDA. Canada has the CHA. Europe has the EMA. But the US has the gold standard. The FDA requires that drugs, devices, interventions be both safe and effective. And so we do a lot of work at the intersection of research and regulatory affairs, educating the FDA about current advancements, talking to them about what a clinical trial might look like, because although there's 8.5 to 9 million people with type 1 around the world, there's not as many people with type 1 as there are with, say, type 2. So if we have a drug or device or treatment that we want to test, it's really not feasible for us to find maybe 6,000 people who have antibodies for diabetes, but type 1 diabetes, but don't have the disease yet. I mean, that's just a bar we can't reach. So we work really closely with the FDA and with other academic programs to figure out the most effective, efficient way to get trials going and get them through the FDA to ensure safe and effective drugs, devices, and treatments. Another piece of ensuring that we reach the people we love or ourselves is health policy. And for us, health policy means accessibility, whether it's through insurance or whether it's through some other mechanism. So amongst the things that we do for accessibility as a part of our advocacy efforts, we hold government days in DC. We hold children's congresses in DC. We meet in our district with our representatives and senators to talk to them about what type 1's like. We talk to them about funding for research through the NIH, and we talk to them about accessibility issues. Many of you probably are aware that there used to be a barrier to access for CGMs, continuous glucose monitors, in Medicare. And it is definitely a very big point of pride for JDRF that we played a role in getting that changed so that Medicare would allow for people on Medicare to use CGMs because people were going from regular insurance where they had CGM to Medicare where they couldn't get it. So health policy is meeting with insurance companies and with legislatures to address access. Many of you have heard of the Insulin Act, right? So that's one of our advocacy efforts in the area of accessibility and health policy. Another thing I'll just mention in terms of accessibility, because again, it's a really big point of pride, is that JDRF invested alongside other nonprofit organizations and philanthropists in the manufacturing and bottling of generic insulins. We should be up and running by the end of next year, if all goes as expected and anticipated, is in partnership with a nonprofit company called CivicRx. The manufacturing will take place for right now outside the U.S., but the bottling and packaging will be in the U.S., in Virginia. I got to tour the plant. If you want to see a picture of me with a hairnet and lab coat and shoe covers, it's pretty funny. But this insulin will be priced at $35 a vial and $55 for a box of pens, making it accessible to anybody, whether they have type 1 or type 2. While we're waiting for Congress to figure out their legislative action, this is what we're doing, and we feel it's really important. The last piece of our pipeline, going from bench to bedside, is what we call adoption. And so adoption can mean a lot of things. It can mean making sure that the standard of care guidelines that are set forth by the American Diabetes Association include a new drug, a new device as the standard treatment for type 1 diabetes. It can include making sure that clinicians are educated about what a new device can do. One of our JDRF chapter board members from right here in Cleveland, Julia Blanchette, where are you? Because there's a lot of light here and I can't see her. Oh, she's okay. I'll tell you what Julia's doing right now. Julia is a nurse practitioner, PhD, who works here at Case. She's in an endocrinology clinic, and right now she is training clinicians in Hawaii on how to use the Omnipod automated insulin delivery device or artificial pancreas. She is training them so that they can help their patients adopt this technology. So really very thoughtful pipeline from bench to bedside. Of course, our roots are in research, and that's where we spend most of our time. And the way we develop our JDRF research strategy is, first of all, we have in-house bench strength in the form of probably a dozen PhDs who work in our research department. And they are probably no more than 1 to 2 steps away from anybody doing type 1 diabetes research in industry or academia in the world. And they develop, in consult with experts in each area of research, a research strategy. We then take a research strategy and we bring it in front of key opinion leaders who are on our strategic advisory panel. Those key opinion leaders have been in industry, they've worked in regulatory affairs, they've worked in academia, they've worked in clinical practice at the point of implementation, and they tell us whether there's any gaps in our strategy that we aren't addressing. And so once we go through this rigorous process, very thoughtful process of developing a strategy, the staff at JDRF Research brings the strategy to the research committee, and we discuss, evaluate, and then ultimately approve. So it is a living, breathing strategy. It is designed not to be all things to all people. We very much focus on gaps and barriers to making advances across what we have set out as a roadmap or target product profiles in each area of our strategy. And we have a set of impact metrics that our staff partners apply on a regular basis to make sure it remains relevant and lives and grows. So our strategy right now is focused on 2 big buckets. Those buckets are cures and improving lives. So in cures, we have 3 main, I'll just say buckets again. The first bucket is screening, because we believe that ultimately universal screening is going to allow us to prevent T1D altogether. The second bucket, and let me just say for a second, these buckets in cures and improving lives, they very much align with the stages of T1D. Does anybody know what the stages of T1D are kind of like? Maybe. Julia would know. Julia is also a research information volunteer, so she goes out and talks a lot about the JDRF research strategy and portfolio as well. We call stage 1 type 1 diabetes when the antibodies have started forming. Stage 2 type 1 diabetes is when the antibodies to the insulin-producing cells have not only started forming, you're starting to see symptoms that are subclinical, so still in the blood, of changes in blood glucose or dysglycemia. Stage 3 is when people are diagnosed. That's when they have open, overt clinical symptoms. And so screening for T1D antibodies, we believe ultimately will lead to preventing the disease for people in stage 1. And right now, screening allows us to find people who are a candidate for a newly introduced drug called Tzield, which delays the onset of further clinical symptoms. So, and then the other part of our portfolio in cures screening, the other parts are disease-modifying therapies. That's where we would change the immune response. That's what we are doing in stage 2 with Tzield, or rebalance the immune system, or we might cause the beta cells that are still in your body to be less stressed and function a little bit better. Or maybe ultimately, and this is still in discovery science phases, basic science phases, maybe the existing beta cells or other cells in your pancreas could be caused to regenerate and make more insulin on their own. Because we really feel like the internal biological solution that's closest to how people without type 1 diabetes work is really important. So that's our disease-modifying therapies bucket, and that's largely in stage 2 and stage 3. But for people in stage 4, how many people here have had type 1 diabetes or had a loved one with type 1 diabetes for more than 5 years? Right. My family, we are professionals, over 50 years of experience. The biggest part that many of you have heard of from our strategy is called cell replacement therapy. And that would be using cells made in a lab outside the human body and transplanting them into the body like you would do any other organ transplant. However, the FDA doesn't think of it as an organ transplant, which makes it a lot of fun to get through regulatory affairs there. So that's the main parts of our cure strategy. And then what a lot of you are probably really familiar with is our improving lives strategy. And that ranges from faster insulins to medications that might improve metabolic control or reduce the likelihood of complications, to complications therapeutics, to psychosocial interventions and supports, to devices like CGM, continuous ketone monitoring, and artificial pancreas or automated insulin delivery devices. So that's kind of the gamut of our research. Like I said, we very much focus on gaps and barriers because we can't fund everything. And so whenever we have a grant go through the JDRF research department, we have a very thoughtful process of external review where the grant applications are reviewed by experts. There's a chance for the experts to critique, for a rebuttal, critique. A lot of times research might be, narrowed down so it's more focused, or we might take a different approach, or we might make an introduction so that somebody can collaborate. We recently had, I know that sound. I was at tennis yesterday, and one of my friends that I was playing with on another court, his T-Slim went off, and I was like, "Hey, Dale, are you okay?" Like, it's just such an automatic response. Like, I just, I treated him like I would anybody else I know with that device. But anyway, very thoughtful review process for our grants. And we also do a lot of grants that promote collaboration across disciplines because as one of my friends often says, the cell biologists might not be at the same conference as the immunologists, and so they may never get a chance to meet. But we bring them together in consortiums and workshops, and we have grants that promote collaboration. One other kind of grant that we have that I think is really important, and I was telling one of the young women who's was in the lab tour, she's an MD PhD student, we award training grants. So we also award early career grants and later career grants, and those are designed to keep people in endocrinology or maybe bring an immunologist into our field to attract more people to this research that's so important to us. So that's kind of it in a nutshell in regards to the JDRF research strategy and the grant making process. Are we ready to go? Awesome. So there'll be a chance. Oh, thanks. Thank you. There will be a chance for some questions and answers at the end. You're welcome to ask me questions about that. Of course, our team, our JDRF staff team who brought us all together, thank you guys for bringing us all together. We really are grateful. It's always amazing when our community comes together. But they know how to reach me if any of you have questions or want to get a hold of me. And I'm sure Julia would be happy to answer questions too. So without further ado, though, let's bring our panelists up on stage and we'll get started with our panel. As I have already told you, we are so grateful that we have all come together today. Thank you to our staff partners for organizing this wonderful event, bringing together physicians, scientists, volunteers, advocates, and donors, and so many more people. We're so grateful to CASE for hosting the Michigan and Northern Ohio chapter JDRF today on World Diabetes Day. And we really hope that today's presentation inspires you. We want you to remain engaged, and we hope that you walk away feeling hopeful. I was talking with someone in the restroom, and we just agreed. Whenever we go to these events, we walk away feeling hopeful. So that's our hope for today, is that you are inspired and feel hopeful. I already was introduced, and you guys should have received bios for everybody at some point. Yes, yes. So we'll dispense with formal introductions right now and leave the bios to that. And just take a second for each of our panelists to very briefly share their why. And I already did. I am a wife and a mom of T1D, people living with T1D, and I am here because I do not want to have grandchildren with T1D.
Hi, hello everyone. Thanks for coming. This is like a dream come true for me. I'm trying to organize such an event for almost 5, 6, or 7 years. And finally it happened. So I really thank JDRF for making this possible. And each one of you who came here and support this event. The reason I'm doing this is because I was asked by JDRF why I'm doing this. I told them I am scared of needles. And I can't think of these children getting needle pokes 3, 4, 5 times a day. I have a number in the slide whenever I show you. It's a huge number, thousands of needle pokes per year. So I thought anyway I'm doing research, I should focus on type 1 diabetes. And one thing I told one of the meeting I had outside is that these children are getting this disease not at their fault. The nature makes it happen. Unfortunately, somebody gets the disease. It's not like other disease by lifestyle or something like that. So anything possible to make the lives better or get something out there, because we don't have any drugs for this. That's the goal of my aim, but what I'm doing. Thank you.
Can you make sure you state your name as you introduce your why. It's on.
Good evening. My name is Dr. Jamie Wood. I'm a pediatric endocrinologist here at Rainbow. I'm the medical director of our diabetes program here, and I'm also the medical director of our diabetes camp a couple miles outside of downtown Cleveland. My why is I went into the field without really knowing anyone personally with type 1 diabetes, but really fell in love with children who live with type 1 at diabetes camp when I was very early in my training. My why now is that I now have 2 nephews and a niece with stage 1 diabetes. So they're my why now, but I still continue to love to work with kids at camp with type 1 diabetes.
Hi, everyone. My name is Betül Hatipoğlu. I'm an adult endocrinologist and a center director for diabetes and metabolic care at the University Hospital. My why, I am not even sure I am so passionate about type 1D. However, I think it's my grandfather, my mom's father, who died before I was born from type 1D. So I believe that must have influenced my thirst to really cure this disease.
Hello, I'm Rose Gubatosi-Klug. I'm the chief of pediatric endocrinology and diabetes. I work with Jamie here at Rainbow Babies and Children's Hospital. My why started with my dad, who lived with diabetes and had many complications. And so I was a family member who experienced those. This was way before we had all of the modern insulin that we have now. So that inspired me to really focus on prevention of complications, prevention of diabetes, hopefully eventually in type 1 diabetes. And then I loved biochemistry. I was just a nerd, and this is the perfect area for me to be in.
I'm Sarah McLeish. I am a pediatric endocrinologist also at Rainbow Babies and Children's Hospital, and I have lived with type 1 diabetes since I was 3 years old, and I also have had a great experience with diabetes camp. I worked there at our local Camp Homey Nakota when I was in college. That's actually where I first met Dr. Wood when we were both young trainees, and so then I knew that's what I wanted to do for my career as well.
Hi all, my name is Amjad. I'm a pediatric endocrine fellow at Rainbow. My why is seeing on a daily basis with children developing diabetes and the daily struggles that children face with diabetes. I feel like we need to continue research and continue developing more therapeutic options so that they can continue to have a happy and healthy life.
Hi, everyone. My name is Corinne. I'm an MD PhD student here at Case Western, and I work in Param Ramakrishnan's lab. So I think my why is I've always been very interested in cellular metabolism, and then I've also been interested in pediatric endocrinology just from my childhood. And so when I was looking for a research lab, I was really lucky to find Parm's lab, his lab really meshes those 2 interests and allows me to research type 1 diabetes. And as a MD PhD student, I'm hopeful that the lab work I'm doing today could potentially help my future patients as a pediatric endocrinologist.
What an amazing group of very dedicated, committed, passionate, professional clinician, physician, scientists. Thank you so much. So as I mentioned before, as part of our cures strategy, JDRF focuses on research in disease-modifying therapies. And so it's now my pleasure to bring up Param. Say your last name again.
Param Ramakrishnan.
I can do that. Ramakrishnan. Param Ramakrishnan to tell us a little bit about the work he's doing in disease-modifying therapies. Just a second, sorry. Just making sure I do my transition correctly. So he's going to explain a little bit about novel targets as therapeutics to either prevent, delay, or reverse the course of the disease. And he'll give us some explanation of what he means by novel targets versus something that might be a systemic immune modifying therapy. So, Aaron?
So hello everyone. I'm trying to show what we are trying to do in the lab in very simple manner. So I'm not going to show you any complicated scientific data or anything that you won't be able to understand without referring a textbook or something. So everything is diluted in a way that you just look at it, you understand. That's what I tried. So this is the first event. As I mentioned, this is the first event in case that it is happening in conjunction with support from JDRF. So thanks again for that. Before this event, we also had a behind-the-scene lab tour for a selected number of people here. So my students, they took them to the lab and showed what is happening in the lab exactly. You can correlate when I talk about things that everything was generated in the lab upstairs. So this one, I picked this slide 10 years ago. It says, let's say, desperate echoes for a therapeutic for type 1 diabetes. Who can say they didn't hear this? When such things are happening out there, we have to open our ears and see something is there. There is a problem. We have to do something about it. That's the feeling I got. Just give 10 seconds to read. And the interesting thing is, since 1920 till to date, insulin is the only proven treatment available for type 1 diabetes. That's really surprising. If you take any other disease that affects mankind, there is some kind of treatment coming up in 5 years, 10 years, 20 years, or something like that. If you call for a medication or a drug to treat type 1 diabetes, as far as I know, till to date, nothing exists there. There are certain type of therapeutic exists there. I'm not saying that nothing is good or nothing is the best or something like that, but you don't have a medication. Insulin is the treatment is there, but if you think loudly, it is not a drug. It is something, it's a hormone missing in your body that you are supplementing it back there. It's a chemical that is missing in the body, you're putting it back. It's not a drug or treatment that somebody developed to treat. And this is the number that I was trying it in the beginning. This is the rough calculation I made manually based on an average insulin injection. So a child with type 1 diabetes is getting 2190 finger pricks and 1463 insulin injections per year. This is a huge number. And the life of these children is like, mostly kids go with one backpack. These kids go with two backpacks. One is the supplies to stay alive. So now I'll try to borrow a word from Walt Disney. It's a very powerful word. Imagine. When you start imagining, there is no end. You can achieve anything. So here the point of imagination is having an oral medication. The most child-friendly way of treatment would dramatically improve the lives of these children. So that is my fundamental goal in this research area, to get something that you can take as a pill and try to treat the disease. Insulin is there for over 100 years, but it also has its own complications and problems. So this number is scary. If you look into the bottom, most of you know these complications more than me, but about 300,000 emergency room visits happens from misfired insulin or hypoglycemia. This 300,000 number, this is an actual number. If you Google it, you can find it. This 300,000 number is more than the number of incidents of disease that's like 200,000 plus there. So there is more complications from insulin than the benefits. And this affects every major organ of the body. The hyperglycemia, if it is not managed well, ups and downs of hyperglycemia, so slowly it can affect each and every organ in the body, and this can actually decrease the life expectancy by 20%. And the rate is increasing in the US. There is a significant increase of rate of about 25% in the last 10 years, and about 20,000 children are affected in the US every year. 50 kids are diagnosed per day in the US with type 1 diabetes. It's an interesting number because 50 states in the USA. Every state, if you can convert the 50 number, every state has at least one child diagnosed with the disease every day. That's how the number piles up. And CDC calculated this and they have predicted this number In 2017, it was about 200,000. They predict in 2050, by 2050 it's going to cross 600,000. And the cost associated with this increasing from 15 billion to 45 billion. There are therapies out there. Most of this, we heard about this IL-2 combination therapy. It eventually leads to beta cell destruction. So it was not, it's not in practice now. And the therapies using CD3, CD20 antibodies exist there. And people think that this can give transient protection, not a permanent solution. And T regulatory cell therapy, it also offers like transient protection, but people think that it can cause delayed autoimmunity. That means further killing of beta cells. We mentioned briefly about beta cell replacement therapy. This is a funny thing here. If you look closely here, the islet transplantation is approved only for patients above 18 years old. And the disease happens in children. And most of the diagnosis cases are under 18 for type 1 diabetes or juvenile diabetes. So that means they have to wait till they become 18, go through all the disease processes, and then wait for a donor to get the matching pancreas, and then always immunosuppression. You have to suppress immune system to avoid the rejection. So it has its own complication. That means there is an unmet need for a novel approaches or control autoimmunity to treat type 1 diabetes. So what I mean by novel is simply new, something new. We need something new that is not out there. And we need novel target, because if there is a problem, we have to find what is the problem, what is the root cause of the problem, and then treat that root cause, not a systemic treatment. So just for an example, we have a problem of leaking tap, right? One option is option 2. Close the main water supply. Leaking tap is gone. Problem is solved. But what is the actual thing that we have to do is you have to fix the tap. You don't have to shut down the water supply for the entire house. Find the leaking tap, fix it. Why this is important is because the problem here is the bad T cells. T cells that kill your own beta cells. That is a problem here. And the therapeutics out there, like T cell blocking therapeutics, they block T cells. All the T cells, good ones, bad ones. They put a wide net and block all the T cells so that T cells are not going and killing your beta cells. And this will lead to poor immunity. If you had heard about the COVID time infections and all, those who are recovering from COVID infection, they have good T cell function. You need T cells to prevent the infection by viruses. T cells are needed to protect from cancer. So if you block all the T cells, you become immunodeficient. So you don't want that method forever to suppress your immune cells in the body. So what we are trying to do here is block only the bad T cells, leave the good T cells there, so that the normal function is not affected. If you have a virus infection, you have T cells to fight against them, but the bad T cells that kill your beta cells are blocked here. So that is a targeted approach. That's the word what we mean by target. Target the specific thing that you want to block, not the systemically block everything, put a wide net and block all the T cells so that then nothing happens there. So that it comes with this, you have to pay off. Poor immunity and probably a cancer later in life. Similarly, here the problem is an infected plant, right? Option 2 is you go to Home Depot and get a bunch of plants and plant more there. What will happen? The infection is there. This is going to infect everything. All the plants will be getting infected. So the solution here, what you have to do is find the plant that has a problem, get the pesticide or something and specifically treat that problem, not planting excess because the problem is there. So you have to treat the specific problem, then you can go and plant more if you need. Why this is important? This is what happens in beta cell therapy. If you put more beta cells, when the patient has autoimmunity going on, T cells are killing beta cells. If you're supplementing with beta cell therapy, the root problem is there. The T cells are going to kill these beta cells. And unless you block these T cells, all the cells that you supply, eventually they are going to die. That is why they are putting again the wide net on these patients. They put the beta cells and block all the T cells again. That means again they are immunodeficient or immunocompromised. They don't have enough good T cells to fight against infection or protect from cancer. So here also we do the specific approach, block only the bad T cells, and this is useful to protect your own beta cells as well as the cells in case somebody gets a donor and get the beta cells to protect also those cells. So you have to block the root cause, that is the bad T cell function. So that's what we are trying to do. And for this, what we are trying to do is we have a protein called CREL in the body, and that CREL protein is very important to support T cell function in general. So the protein is really fair. It supports bad T cell, good T cell, it doesn't matter. If there is a T cell, this protein will go and support it for them to grow and survive. So what happens in diabetes is that when the serial protein, when there is hyperglycemia or more sugar there, the protein gets modified by sugar. So when the protein is modified by sugar, what happens there is they have a selective support for bad T cells over the good T cells. So when the sugar is slightly increasing, the early stages of the disease, the protein gets sugar, they become hyperactive and they support the bad T cells. And these T cells go and kill the beta cells. And this goes like a vicious cycle. More sugar, more damage, more active protein killing of beta cells. So this continues, this process continues. So that is a self-accelerated process of killing beta cells. So for this, what we are trying to do is, now we know, now we have a target, right? This is the target we talked about, a specific target. There is a protein called serral. The protein is modified by the sugar, and the sugar modification is the problem here. So we have to block it. So for this, we collaborated with chemists and who did the computational designing and modeling, and they came up with drug models. And this drug will bind only to the sugar modified protein, not the normal protein. So going back to my first explanation, you kill only the bad T cells or you block only the bad T cells, not affecting the normal cells that are required for your normal health and survival. So we made these molecules with these two faculties. He was in Case Western, he moved to UCSD now, John and Kent. Kent is also supported partially by JDRF funding that I received. So we have this drug, and what happens is once you put this drug, it blocks the sugar modified serral. The function of bad T cells go down, the good T cells take the upper hand, and you have a healthy pancreas that can produce insulin. And if this happens, in reality, we have the cellular experiments at the bench. We see that this is happening. At this stage, we are trying to make an animal model to put this into the animal and see what is going on. And if this really happens, type 1 diabetes is in control. And this is the drug that we have. And it actually, based on the evidences that we have, we can call it as a drug, that it works as expected. So our advantages, I think it's partially cut off. Advantages is that, as I told, it blocks only the disease-specific modification of the protein, not the normal health or not the protein that is required for normal health. So just to give an outline of what is happening here, the green is where we stand now. Year 1 is completed with support from JDRF, and we are trying to do the mouse studies now. So the mouse studies and preclinical studies will be completed in next 2-3 years. And optimistically, if everything goes well, we will be in 5 to 6 years of time. We believe that we can enter clinical trials. So that was the part 1 of the story that leads us to a successful outcome, an oral medication here. So from this, you can escape to have a medication that you can swallow and feel better. So this will be like the non-invasive, first of its kind oral medication. This is what I told, something is lacking in the field. You don't have a tablet you can eat for this disease. The second story, Corinne, my student, will be explaining about this. So we have, that one I explained is a protein therapeutic. And nowadays, people do RNA therapeutics. So we have a second project that we work on RNA therapeutics. We find some RNA that has a role in type 1 diabetes. And this is our dream model. This is the normal mouse that some of you saw upstairs. When they become sick, they look like this. Once we put our therapeutic, this is going to become normal again. And we have some indications that she will be explaining in her presentation. And again, as Disney mentioned, if this happens, we can tell it all started with this mouse. And thanks for JDRF. I would have one more thing. Finally, I would like to thank JDRF for the opportunity. And if Elizabeth was asking me what this means to me, this funding, what this funding means to me, or how this helped me. So that I would like to show that without this funding, what would have happened is just this. You won't see anything. Blank, no data, no presentation. We won't be able to do anything what we are doing now. So I'm really thankful for the support that made all this possible here. And thanks to my lab research team, all the people present in the lab now, previous people like George, Tristan, Jeffrey, all of them contributed to this project. And my 2 chemist collaborators, Ken from New York University and Jonathan from Case Western. Other collaborators at Case Western, Tim Kern and Rashmi Parmeswaran. And my mentor, David Baltimore, where I started all this work. and all the collaborators in UH, and these people are here in the panel. So thank you for being here and supporting this work, specifically Sarah, Rose, Betul, and Jamie. Thank you very much. And the previous collaborators from UH. These people left the institute, but they all helped me to go. And thank you all. Yeah.
Before you step down from the stage, I do have one question. You guys might remember that I talked about our JDRF Cures portfolio as including screening, disease-modifying therapies, and cell replacement. Clearly, this falls strongly in the disease modification space. Param, how do you feel like screening for autoantibodies and detecting the disease earlier interfaces with the type of work you're doing? Because it seems to me like you would want to catch someone really early in order to use this oral therapeutic before they progress to full-on clinical disease. Is that true?
Excellent question. Now I know who reviewed my grant.
Not me.
Yeah, they asked the same question. How you can put it, when you put it in the clinic, how you can utilize this to treat early people. That's what exactly we want. Because many times this runs in the family. We know the genetic background. We know the autoantibodies. So we know who is going to get this problem. A slight increase in sugar is going to activate the protein and cause a rapid amplification or rapid progression of the disease. So in case you know a sibling is getting the disease, the next one may or may not get it, but still it's totally fine to put them in this treatment because our data shows that this is not blocking the normal function of the protein. It specifically affects only the sugar-modified hyperactive protein. So optimistically, there won't be any big complications or side effect by starting this treatment early and having the knowledge from autoantibodies or some other, if the disease runs in the family, it's totally fine to consult with the doctor and ask, is it okay to take this kind of medications?
So another example of why screening is so crucial to preventing or delaying onset of this disease. Thank you so much.
Thank you.
We already brought it up. Thank you. So next, I'm going to bring up Dr. Jamie Wood. And I mentioned a little while ago our Improving Lives portfolio. And Dr. Wood, if you would share with us a little bit more about why the insulins that are commonly used, most of us probably know of, or have a connection with a fast-acting insulin or a long-acting insulin if you're on multiple daily injections, why those really aren't like quite sufficient to do the job that we would like them to do in helping manage blood glucose.
I actually sort of answer that question in my talk. So I'm just going to jump into the talk, but thank you for the invitation. And as was mentioned, my work really falls into the CARES department in that while we're waiting for the cure, we need to take really good care. And we really need to improve the lives of youth and adults living with type 1 diabetes. In my work, I do lots of different type of clinical research in the clinic. And this this inhaled insulin trial that I'm doing is one example of trying to increase the options available to people living with diabetes to manage their diabetes. And it is World Diabetes Day, so we're celebrating Dr. Banstein's birthday. And you can see just how far we've come between now and then with glucose monitors and CGMs and pumps and automated insulin delivery. I mean, it's a pretty exciting time to be working in the field of diabetes and caring for people with diabetes because we have more and more options to improve lives. But it's still, the burden is way too high. And these are two statistics that we talk a lot about. And there's one estimation that if you live 24 hours with type 1, that you are making, on average, 180 decisions each day about your diabetes. And this other study on the left estimates how much time of a day is spent thinking about diabetes. Now, I think many people living with diabetes or carers of people living with diabetes would say it's more, but children on average spent in this one study an hour a day doing treatment-related tasks, not just thinking about diabetes, but actually just doing treatment-related tasks. So the burden is still too high. So we need better ways to manage diabetes while we're waiting for that cure. And we also know that young adults, particularly teenagers in particular, are not achieving the glycemic outcomes that have been demonstrated to stay healthy. And this data actually has been recently updated. I don't have it in this slide. It's getting a little bit better, perhaps. But the orange line was several years ago, and this is the A1C across the lifespan. And you can see where youth in diabetes are right here. Right, and so the A1Cs are much higher in youth for many reasons. And the blue line was the most recent data published in 2019, actually showing despite all of these advances, the A1Cs were actually a little bit higher. So despite this great technology, we still have a lot of work to do. And one of the problems is the question that I was asked is we need faster acting insulins. And if you think about injecting insulin, or you think about insulin in pumps and automated insulin delivery systems, they're not human. I mean, they're human insulin, but they're not like the insulin that is made by the beta cell in terms of how fast they are. And this schematic I borrowed shows how the insulin that we're using now here, so this is a rapid-acting analog like Humalog, Novolog, Apidra, one of the rapid-acting, with where it's working its fastest at about an hour. It's working its most at about an hour after it's injected, and then stays around for 3, 4, 6 hours in some people. But the insulin that the pancreas makes is instantaneous and is peaking. That's in the dotted yellow line. It is much faster and can dissipate more quickly. And then inhaled insulin, which we're going to talk about, is even faster than the injected insulins. And one of the limitations to getting the glycemic management where people want it is in how fast this insulin works and how long the insulin sticks around. So inhaled insulin was approved by the FDA for adults with type 1 and type 2 diabetes in 2014. And it is an inhaler. It comes with this inhaler here. And then it comes with 4-unit, 8-unit, and 12-unit cartridges that are popped in and are inhaled. And so someone with diabetes would stays on a basal insulin injected, and then uses the inhaled insulin at mealtime and to correct high sugars. And how it works, I'm not going to say the word. It's as FDKP, and so it's this technosphere, and so this microparticle is combined with insulin in that product. That combination is called Afrezza, or technosphere insulin, and when it's at acidic pH, it's clumped into particles, but when it's inhaled, the body has a more basic pH, a physiologic pH, and so the particles dissolve, and the insulin is released into the circulation within minutes. And so this is a comparison of inhaled insulin versus injected insulin. And you can see that the inhaled insulin, as I was mentioned, is absorbed to the bloodstream within minutes, and then is its strongest at about very quickly, and then is dissipating and almost out of the system at the hour to hour and a half mark, versus injected insulin is not reaching its peak until 60 minutes and then can stay much longer. So the risks and benefits, there's some cough and some throat irritation, and people who have lung disease, have, COPD or asthma, or who are smokers, shouldn't be taking it. And in adults, there was some more high blood sugars when people first started on it and were getting used to the dosing. There was some worry about lung cancer in the adult studies, but of the 3,000 people that were studied, 2 people developed lung cancer. smoker, and both those two people were very long-term heavy tobacco smokers. So probably not related to their use of Afrezza. But the benefits are that you can manage your diabetes with just your basal injections, not having to inject multiple times for meals, may improve management. The studies in adults show that the A1Cs are comparable with fewer lows. And what's nice about the inhaled insulin is that you can correct highs much more quickly so that the high blood sugars can come back to target faster. And that allows you to have less stacking of insulin. So you give insulin for a high, and then you're taught, well, you can't give another injection for 3 hours or so after the previous one because the insulin doses might stack up. But with Afrezza, it's really out of your system, and you can dose more closely together. We also find that exercise is easier with Afrezza because, like I said, you can dose for a meal and then it's out of your system more quickly before exercise. So what we're doing here at Rainbow is this is a pharmaceutical-sponsored trial. So Mankind makes Afrezza and is sponsoring the trial, but this is regulated by the FDA. And so we're taking roughly 300 kids, children under the age of 18, both type 1 and type 2, down to the age of 4, and randomizing them. Half of them get Afrezza and half of them stay on injected insulin. Both still are in their basal. We do that for 26 weeks. And then after 26 weeks, everyone gets Afrezza for the next 26 weeks. And we're really looking at our primary outcomes. So the most important thing we're looking at is that A1C at that 26-week mark. And we want to demonstrate that Afrezza is as efficacious as injected insulin after 26 weeks. And then we also want to look at lows and highs and satisfaction with the treatment. And those are sort of the secondary objectives. But the main thing is that we also have to prove that Afrezza is safe in kids and young people with diabetes. And just because it was safe in adults does not necessarily mean it will be safe in kids, and is inhaling insulin safe for the developing lung. And we feel fairly confident about the safety in adults, but we have to demonstrate that in kids. So that's what our study's doing. And we're looking at these safety endpoints, primarily pulmonary function, as well as how often do they have bronchospasm, like wheezing, any hypersensitivity reaction. This is just a demo of how to use it. It comes with this inhaler, and the cartridge pops in, and you inhale it forcefully and quickly. And then you can sort of pop out the cartridge, and kids combine these cartridges to get the dose they need. 4 units of inhaled insulin is equivalent to 2 units of injected. So I have teenagers in my trial who are inhaling like 52 units. And it sounds crazy, but it is. But it's like, that's like 20 units of injected, but big doses. So you have to bind cartridges and do cartridges one after the other. So I actually have a multi-center call tomorrow, but our goal is 300 pediatric participants. And there's, I think, close to 50 now centers and we're working together to do this. And so we hope to be finished enrollment by the end of December. I'm enrolling our 7th participant tomorrow and my 8th participant on Friday. And so here at Rainbow, we're going to be up to 8. And this is one of our little guys. He's our youngest, and we call him JT. And he's pretty phenomenal. And our research nurse, Paul, our research nurse, Terry, and this is Mom and Dad. And my other message today is that participating in clinical research is a lot of fun. I love working with these families and that do research with us, but they also, I think, get a lot out of it too. They have an opportunity to try a new therapeutic before it's available. And then they also get to be part of something that's bigger than just their family. And the opportunity to give back and help the diabetes community means a lot to these families. So we want to thank JT and his parents. And here you can't see it. His inhaler is kind of covering up by his hand, but he's practicing how to inhale insulin here at our research center. So in conclusion, I feel very strongly that people with diabetes need options. Not everyone wants a pump. Not everybody wants to wear a CGM. And we have multiple different pumps and CGM combinations and options, and those options are really important. And I feel that inhaled insulin is just another option. It's not going to be right for everybody, but our families living with diabetes deserve to have as many options as possible out there. So hopefully we'll be able to demonstrate that inhaled insulin is safe, primarily and first and foremost, but also efficacious in children. And then come visit us at Camp Homey, Dakota. This is my favorite place. I'm the medical director, and as Dr. McLeish said, we met there very early in our training days. So come visit us if you ever want to. I think I've got my camp friends up there in the back. So come see us at Camp Ho.
So thank you so much, Dr. Wood. I really appreciate it. You're gonna move us to the next slides. Thanks so much. How many of you had heard of inhaled insulin or Afrezza before? So a couple, for sure. Remember when I was mentioning the regulatory thresholds that drugs have to go through, drugs, treatments, et cetera. I mean, that's a really great example of the bar that the FDA sets for us in terms of efficacy and safety. And it's really exciting that this will be an option, presumably be an option for kids. And I will also just put one more plug in for clinical trial participation. And if you are interested, JDRF has a clinical trial connections tool, and within 60 seconds, you can find trials you might be eligible for. Now it's my pleasure to bring up Dr. Betul Hadipoglu. Did I get it right?
Perfect. Excellent.
Oh my gosh. So as we mentioned earlier, cell replacement therapy is one of the JDRF strategic priorities. And Dr. Hadipoglu is going to talk to us a little bit about how islet cells work, and what islet cell replacement is like, and what the opportunities in this area of research are.
Thank you so much. I would like to thank JDRF and Param. Thank you. I have my fans already. It's always good. So it has been absolutely a pleasure, and it was by total coincidence, if you believe in coincidences in life, to really find myself in the middle of islet cell work. This is my grandfather, which I have never met. He is with his own father. Both were surgeons, general surgeons. I was born and raised in Istanbul until I came to US 30 years ago. And he was found, unfortunately, unresponsive in his office, which we believe must have been an insulin reaction. We are talking about 1960s, when they didn't have a whole lot. No options. My oldest patient, believe it or not, he's 89 years old, type 1 diabetic, since age 5. I just made a video, if I can get it through UH's regulatories. I'm going to put it on my website before the poor guy, something happens to him. I told Don, you're not allowed to die under my watch. So he's so compliant with his visits. He always comes. He's such an inspiration, such an amazing person. We helped him. UH sponsored one of his books that tells all these stories. You know, some people, they look at everything so different. You talk to them, they might suffer like rest of us, but they bring in this joy. If there is a joy in a suffering, he brings in that. And he brings that in for others, like young people, like teenagers, like audiences. When he talks, you know, that flame of hope that we just talked about just comes back in people's. We need people like that. For type 1 diabetes, for diabetes in general, which is a pandemic. So severe hypoglycemia and its effect has been somewhat my passion, because thinking that I could have met my grandfather. We do have in individuals who fails normal treatment options currently, possibility of giving them pancreas transplant. It does work. It helps survival, and it does help patients to be insulin independent for a while. But you need immunosuppressive drugs. And when Param says, this is not approved for 18 years and below, I would say, don't touch my kids. When the transplant surgeons were like, yeah, I need to try this on kids, I'm like, no, you're not touching them. Because immunosuppressive drugs, we don't have something that is safe. And I evaluate people for pancreas and islet cell transplant. When I tell them you are not a good candidate, they think I'm taking away the best thing on earth from them. And I tell them, I am protecting you. This is for you that I'm not approving this. Because nothing happens to me. It will be you, your precious life. So around, I don't want to talk too much. When I find a good audience, I love talking. So around the time I finished my fellowship, there was this publication about islet cell transplantation. Islet cell transplantation, islets, which are the pieces of the pancreas, 1%. 1% of the whole pancreas is islet. 99% belongs to GI guys, gastroenterology. 1% is mine. And that 1% has these beautiful looking cells that controls the sugars. And you could actually just bring them out and give it to the patient. You don't need the whole pancreas. Why? The whole pancreas transplant is a big surgery. And not everybody, like if Don needs it, he can't have it because it's too much risk for him. So islet cell transplant, the idea was there since 1960s. However, it wasn't possible because we didn't have good immunosuppressive drugs. So the technology improves, immunosuppressive drugs improves. So in 2000, there was this amazing publication that they were so successful with islet cell transplant. So I happened to be at the right time, at the right place, just a coincidence, that totally helped me to build my career. It was something I just loved. So I helped the University of Illinois team as a clinical piece for them, work with this amazing group, and JDRF actually helped them as well at those years. I'm talking 20 years ago almost. And they were in the lab. This is a huge collaboration. Param, us, you, this is a team work. JDRF, we all have a piece to play. Research and innovation is collaboration. So the team would get the pancreas, just digested. This will be this junky looking material with a lot of exocrine, those 99% we don't want. And this is 1% I want. And then it's given into the liver. So we did it, and I first saw a first patient in front of me receive it. And within 24 hours, not needing insulin and sobbing. And me, I'm so filled with empathy for my own goodness. I cry with her. We cry. We're like hugging each other. We're just in tears. So powerful. A resurrection in front of you. It's just amazing. So I was a part of this team. Tedious work over and over, finding the right patients, giving it, making sure it works. When it doesn't work, you are as crushed as they are. The islet cell transplant team, with my team, and we had some different protocols. I don't want to take too much time. I know we have an amazing team of other panelists who are going to come. But what I want to say, we published, we did research, we followed our colleagues. You know what I mean? It was boring because we were moving. But this is a difference between transplant surgeon and an endocrinologist. I was working with Jose Obroholzer, God bless him. The guy is so like type A. Even if I am type A, he's like double type A. So we quit, like something goes wrong. I'm like, oh, went wrong. He's like, so I say, if someone can open a hole to the wall, he will do it. He just pushes. So we did it. I'm going to just come here because I want you to know each research might not be what I have seen. And I have seen the starting of a project, the collaboration, and I have seen now this year an approval by FDA, the same product to be used for type 1 diabetics. I cannot tell you how much that means to me. It was the hard work nights and days of teams that worked in the labs, of clinicians that worked with them, our patients who volunteers their life for this, and all these agencies like JDRF, NIH, pouring support, because without their support, we couldn't have done it. So the Donis-Lacelle is the product. We worked together with the team. Someone once told me, one of my mentors, that it was sacred to be involved in the start of something new. And I still feel that sacredness in this islet cell transplant. Yes, it's not for everybody. Yes, however, we need hope for everybody. And this is a hope. I will just close about a recent research study that I am trying to get at UH. It's a phase 1 trial, never tried before. It's a neuropeptide to administer it to the pancreas. Per the company and their animal studies, it actually reverses type 1. Too good to be true. We will see if it works in humans. And this might need something like Param's, what he's working, because this doesn't affect immune system. There's no immunomodulation. There's no immunosuppression. So we might need to have something in addition to the product if this works. So it's on the, I might call you. Thank you.
Thank you so much for being a part of the very long history of arriving at a point where the U.S. approved donor islet cell transplants just late this summer. Thank you. The work of you and many, Dr. Osterhoser, is really important. It is a very important first step also to have this donor islet cell transplant approved by the FDA, because this product met the safety and effectiveness protocols the thresholds that the FDA required. And so what it does for us, as was mentioned, this isn't going to be for everybody. I mean, there's just simply not enough donor islet cells. And it does still require immunosuppression. So no children, that's just not even feasible. But what it does is it lays the groundwork for when we can generate cells in the laboratories for that kind of cellular use to get approved by the FDA. So it's really, really important. It's a great option right now for certain people. It does bring us hope, and it lays so much of a path that we can follow for stem cell-derived beta cells that are made in a lab. So now I'm going to bring Dr. Gubitosi King to share a little bit about her work in she's a clinician scientist, both in the lab and in the clinic in regards to complications.
Thank you. Thank you for the invitation to speak, Param, and thank you, JDRF. And so, as Dr. Hatipoglu was saying, 2023 is a really remarkable year. It's the first year where we have an FDA-approved drug to slow the onset of type 1. And it's also the 40th anniversary of a landmark study that really taught the world and taught all of us about intensive diabetes management. It's really been the study that's been the foundation for hope for all of the new therapies and all of the exciting information that we've just heard about. So I want to tell you a little bit about the study. I've had the opportunity to be involved for about the past 17 years in what's called the DCCT edict, or the Diabetes Control and Complications Trial, and then the long-term follow-up of this same cohort in the epidemiology of diabetes interventions and complications. So going back, a history lesson, we've talked some history. 40 years ago, the Steno Hospital in Copenhagen looked at 40-year-olds with type 1 diabetes, and there were staggering rates of complications to the eye, the kidney, the heart, and the brain. And morbidity and mortality was increased 2 to 6-fold compared with age-matched individuals without diabetes. And this was really what led to the Diabetes Control and Complications Trial. Was we didn't know back then, was glucose really what was causing all of these complications? And if we were able to regulate glucose levels to near normal, could we actually delay or prevent the onset of complications of diabetes? And so I think it's important. We've talked about public policy and impacting public policy. This was actually in the United States a directive from the National Commission that said we have to study the complications of diabetes and understand what's causing these rates of complications in type 1 diabetes. And so as Elizabeth explained, the NIH released a request for applications, and there was a planning phase that led to the Diabetes Control and Complications Trial. So a big study like this takes a long time to plan. It had two arms, one that we were looking at primary prevention, so individuals with diabetes type 1 that had very short duration, had no complications, and then another group, a secondary intervention, because there were many individuals living with type 1 diabetes. And so those with a little longer duration who had some early complications, could we prevent the complications in the primary arm, and could we slow the progression of complications in those already experiencing the early complications? And so the interventions, which seem really funny to us nowadays, conventional therapy at the time, and this was, we were talking a little bit about the glucose sensors and how big they were, was really only 1 or 2 daily injections of insulin. Monitoring was done by urine. There were no glucose monitors. And the goal was no symptoms of high or low, and A1C under 13% was thought to be where it needed to be safe. For intensive therapy, this is where we start the modern day management with at least 3 or more daily injections. And when insulin pumps became available, the use of insulin pumps, and at least for monitoring of just using a standard monitoring blood glucose test. And these were finger sticks when they became available, pre-meal targets, post-meal targets. And the goal was actually to get the hemoglobin A1C under 6.05%. And this was based on some other earlier studies that if we kept it that low, we would prevent the early eye disease. And so recruitment started back in 1983, and then the landmark we had feasibility studies, and the landmark results came out in 1993. So the DCCT lasted for 10 years. 1,441 individuals joined the study. An important aspect was that there were about 189 adolescents, 196 at the start, 13 to 18-year-olds studied. So it gives us some info on adolescents who were diagnosed with diabetes and their course over time. Really representative of the public at that time, about 50-50 women and men, predominantly Caucasian, and again early as well as those with a little longer duration of diabetes. And so what happened with the trial is that they didn't achieve the 6.05%, but those with the intensive treatment on the bottom arm did get down to 7%, and that's why we use that 7% target. And we'll show you more about that in a minute. And those with the conventional treatment arm, just being in the study, kept them around 9%. So you had this 2% separation in their glucose levels, and they're measured by A1C. And that resulted in the major findings from this trial. Enormous reductions in the onset of early eye disease in both the primary prevention, so those with no prior, short duration, no prior history of complications, 76% reduction in the intensive treatment arm, and a 54% reduction in the early eye disease in the secondary intervention arm. So really significant reductions, and it became unethical to continue to use conventional therapy. And so everyone was treated and taught how to do intensive diabetes management. We followed these participants now for 40 years, really 90% of their diabetes duration, and major findings that continue are enormous reductions in all of the early stage complications, and now with further follow-up, when we talk about more advanced stages, eye disease requiring laser or injections, et cetera, intensive diabetes management and HbA1c glucose remains the predominant risk factor. So if you can manage your diabetes well and reach your hemoglobin A1c target, these complications are severely minimized. This is where we really get the 7%, because we looked early on at the relationship of the glucose levels with outcomes. And then we also, as it's been talking about, a huge effort in all of our diabetes clinics is to prevent hypoglycemia. And so you can see at the 7% on the curve on the left that this is on the we're looking at the rate of early eye disease. Once you get down to 7% and you continue to go down, there's very little early eye disease. It really swings up. You have the rate increases above 7%. So again, that's what we were successfully able to achieve in the trial, and it bears out that that's the pivot point for reducing complications. But then on the severe hypoglycemia, using, the insulins at the time and using the monitoring devices we had at the time, 7% was also a point at which below that, you can see the curve swings up the number of severe hypoglycemia events. And so 7% became this balance between we can really prevent the long-term complications and we can minimize as much as possible severe hypoglycemia using the tools at the time. And so that, again, we taught everyone to transition to intensive diabetes management. And then as we continue to follow people over time, you can see that separation of A1C in the DCCT period, that 2%, once we trained everybody, the curves converged. Those with convention treatment dropped, but those with intensive treatment actually rose, because using the tools at the time is very difficult to continue to manage intensively. But you can see as the years have progressed that gradually over time, the hemoglobin A1Cs have improved, and especially over the last 5 or so years where the technology and the accessibility to glucose sensors and automated insulin delivery devices, which have been adopted even in our participants with an average age now of about 60, that this has really helped them to meet their diabetes management goals. This is an interesting phenomenon as well that came from the DCCT edict, something called metabolic memory. And so after the trial ended and we followed through edict, it just maintaining good glucose for any period of time has lasting benefits. So something that your body remembers, good metabolic control, and that continues to show that you can see in the yellow arm that the intensive treatment, even after the trial ended and their A1Cs rose a little bit, they still had significantly less eye disease. So this concept of metabolic memory and trying to understand that at the cellular level has been a big effort from this study as well. And we have some targets. So other findings, again, that glucose control and A1C, that's the primary thing with complications. There are risks to intensive therapy, the hypoglycemia and the weight gain, but not now that we have more sensitive ways of monitoring, we can prevent much of that, but that's why that remains a primary target in any trial is the rate of severe hypoglycemia. There are many beneficial effects of intensive therapy in the adolescents on their something called autonomic neuropathy, their neuropsychological function, their quality of life. So improving A1C improves your quality of life. The effects of intensive therapy also had, and thinking about Batul's work, an effect on beta cell function. And a measure of beta cell function is C-peptide, which is that if you make your own endogenous in your own body insulin, you will make a little bit of this extra C-peptide. It's cleaved off of the insulin. And so that, because of this study, we know that C-peptide is important. Because of the study, we know that even 30 years later, individuals with type 1 diabetes can still have a few functioning beta cells. So it gives you that hope for regeneration. And actually, we compared C-peptide results early in the trial to later in EDIC, and some individuals had slopes that were increasing, suggesting maybe there is regeneration of beta cells that can occur. And maybe it is because of intensive management and good glucose control that is a survival factor in promoting beta cell regeneration. So that's something that we need to study further. And then with intensive therapy, we reduced the morbidity, the mortality. We showed that it had a beneficial economic impact, which is what has helped to get insurance approvals for insulins and additional devices over time. And then we have been following novel markers. So in the field of what additional medications can we add to really completely prevent any risk of complications. And so we're studying those and thinking about new medications to add to the diabetes armamentarium. And then one final result with metabolic memory. Of course, if you start early, so at the top, the conventional treatment group, the bottom, the intensive treatment group with the 9% and the 7%, the earlier you are able to achieve your target A1C, the more risk reduction you'll have, and this is kidney disease. But we recently did some additional analyses that, for the first 10 years, you have an A1C that's above target at 9%. And then for the next decade, you're able to achieve that target. Maybe you changed up your management strategies. There's still a significant reduction. And so I think this is a resounding message of hope that at any stage, if you can improve your diabetes management, you will reduce your progression and your risk for development of complications. And I just have, similar to Jamie, this was really the study that set the pipeline, the domino effect that led to guidelines that changed and led to the explosion of insulin, new types, inhaled insulin, everything down the road. It's really what said we need better technologies. We have to avoid hypoglycemia. And we've gone from urine tests there in the jars to now having these wonderful continuous glucose monitors that help people know at every moment and overnight whether they're trending low. And so, as Jamie, too, mentioned, I just have a few comments. We did a 40th anniversary symposium at the American Diabetes Association meeting, as well as at the European Association for the Study of Diabetes. And our participants sent in their reflections of being a part of a trial now for 40 years. And they were extremely inspiring that because of being part of these studies, they've learned to live life as normal as anyone could hope for. This is their greatest achievement in their life. It's being part of the study and being part of changing the face of diabetes for many generations. New confidence and new challenges and really their lifeline. So thank you for listening, and I'm going to end there.
Thank you so much. I think you heard another pitch for participation in clinical trials. And you also heard a little bit more about what I was kind of describing as our pipeline earlier, where we adopted new standards of care that have led to much better outcomes. And as I mentioned earlier, JDRF continues to work on a portfolio of drugs, devices, and interventions to help mitigate further complications in terms of eye disease, kidney disease, psychosocial impact, easier to control blood glucose, and without the fear of hypoglycemia. So now I'm going to bring up Dr. Sarah McLeish, and she's going to share a little bit about, remember I was talking about in the pipeline about accessibility and reducing some disparities in accessibility. So thank you, Dr. McLeish.
Hello, thank you everybody, and thanks for the invitation to speak. So I'm going to share about a project that we're working on at Rainbow Babies and Children's right now to reduce disparities in technology use for type 1 diabetes. So there are a lot of known disparities in diabetes, in particular racial disparities. So we want to have equal access to our technologies. We have all these amazing continuous glucose monitors and automated insulin delivery systems, and we know they work well, but we don't have all of our patients using them. So just to give you an idea about the overall disparities in pediatric type 1 diabetes care, our non-Hispanic Black patients have 1 to 1.6% higher A1Cs, and in young adults, it's even a larger discrepancy. And DKA rates are much higher in our Black patients, severe hypoglycemia, hospitalization, and risk for retinopathy. It's all a higher risk. So automated insulin delivery systems, probably many of you in here are familiar with those, but as a quick overview, there's 3 components of an automated system: the continuous glucose monitor, the algorithm, which uses that continuous glucose monitor data to calculate how much insulin needs to be given or restricted and suspended, and then the insulin pump that is delivering the insulin based on what the algorithm tells it to do. Right now, of course, there's still a lot of input from the patient and the family as well, but we're getting better and better at making things easier. So on the bottom left there, it's just showing how the system would increase or decrease insulin based on the CGM readings. So all this, we have many automated systems now, more and more coming on the market all the time. Just this year, we've had the iLet come out. Medtronic has a new pump available now. We have the Omnipod 5. We have Control-IQ, and more things are in the pipeline. So it's really an exciting time. All the studies show that glycemic control improves no matter what system you're using, that glycemic control improves. And so on the top right, that is showing the time in range data. For the Control-IQ is the darker turquoise on top, so it's a higher time in range, and the standard insulin pump with a sensor is on the bottom. And then the lower here, where's my, there we go. Sorry about that. So this is showing the time of day. This is a little bit like confusing when you first look at it. It looks like these are glucoses going high, but this is time and range, so higher is better. And it's just showing how overnight, when we're not eating and exercising and doing all the things of normal life, that the automated systems do pretty amazing jobs. And during the day, it's still doing better, but not nearly as good as overnight when we don't have all these other things going on. So another finding of all the studies is that the greatest improvement in control is in those who are starting with the highest A1Cs. So it's helping at all A1C levels. These automated systems are helping, but the biggest change are those who are starting out with the highest A1Cs. So this is just examples of 2 of the automated systems and the studies that went with them. But we have disparities in our technology use too. So on the earlier slide, I showed disparities in A1C and acute complications like DKA, severe hypoglycemia, hospitalizations. But there's also a big difference in who's using the devices. So our Black patients are just not using the devices as often. And these 2 studies were done at the Children's Hospital of Philadelphia, and they were looking at CGM use right after diagnosis. And you can see that CGM initiation was 2.7 times less likely in Black children and 4.1 times less likely to continue CGM use. And then these studies were done in young adults with type 1 diabetes. So you can see that the rates are very different. Ever used a CGM in their life, 28% in Black patients versus 71% 50% in white, and pump 18 versus 72%. And then at Rainbow, we looked at this as well. So we're doing better than a lot of these numbers up here, but there's still a big difference, 72% in our Black patients versus 94% in our white patients. This was actually from 20 up to 2021. I actually think this is probably even it's a higher number now for sure. But, and then ever used a pump shortly after diagnosis, 25% versus 54%. So it's a problem because we need to make sure that everyone is using these technologies, everyone who wants to at least has access. So the study we're working on is to use a remote patient monitoring tool where we gather information from the devices and we're able to triage it. So right now we can look at the data. Like if I think about somebody, like I'm going to look up, Dr. Wood, if she was wearing a CGM, I could look up her data and I could see it, but I have to think about her and go purposely look it up. And we can't do that for 1,000 patients. I can't go look at everybody every day. So this will allow us to set criteria of who we're worried about. Like if you're not wearing your CGM, if you're not staying in automated mode, if your glucose is very high, if your time in range is low, and it'll flag people who are like in a concerning zone. And then we can reach out to them, even if they're not reaching out to us. And then on the patient side, they'll have an app where they can see the same data that we're seeing for them. And they'll get kind of like push notifications, like your glucose has been high for 6 hours. You need to check your ketones. Then they can put the ketone value in there and it will come back up with a message like give your correction dose, or you need to call your doctor. This is high, things like that. And then we'll also have a community health worker. This is really exciting part. We have 2 amazing women who are working as community health workers for us who will be able to go to people's homes and help them as a technology coach. So help them get started, help them troubleshoot things, all the problems that everybody knows that can come up with technology. Like it's amazing when it works, but when it's not working, it's really frustrating. And, if like a lot of patients, it's just not high enough on the list of things to that they can do. I don't have, 30 minutes to spend on hold and then talk to somebody on the phone for another 30 minutes to troubleshoot my device. So we're hoping that the community health worker will be able to help families with things like that and troubleshoot things even with pharmacy and getting supplies. And what if my device isn't sticking, and all the things that can happen with devices. So this slide is just showing our normal diabetes care at Rainbow right now. So someone's diagnosed, and then right after diagnosis, they come for a visit every 3 months, and then we space it out to, at monthly for 3 months, and then we space it out to every 3 months. And these are kind of like the different roles. The medical assistant will get our data for us. The diabetes nurse does a lot of education. The clinician and the nurse will review the glucoses, and we adjust doses if needed. And our social workers kind of touches base with anyone who needs some extra help. But then after the visit, it's really up to the patient and the parent to reach out to us. If they don't reach out to us, we don't know there's a problem, we can't really help. And a lot of people do reach out to us, but a lot of people don't, because again, it might not be the highest thing on the list of the things someone needs to worry about that day. So that's where this comes in. So now, if the patient or parent doesn't reach out to the team, we will have this weekly review of our dashboard. And if they're not in what we call the green zone, where they're meeting goals and targets, then we're going to reach out and contact them and figure out what the problem is and who needs to help. And then as well, we'll have the community health worker visiting them on a monthly basis or at any time as needed. So we did a lot of interviews and focus groups over the past year with our stakeholders. So Black young adults living with type 1 diabetes, Black parents of Black children, and healthcare workers caring for Black children with type 1 diabetes. So these are some of the findings from our interviews and focus groups. So this slide is showing perceived benefits of diabetes technology, which they go along with what everyone would think, but it's kind of what you would think. And then so the color of the box and the size of the box is showing how many people said it. So like this one, less finger pricks and equipment, that's kind of a one that we would probably all say was kind of the most mentioned benefit of the technology. But accessible data, sharing, parents being able to see the data, reducing burden, knowing your numbers all the time, getting the glucose alerts, and then helping to improve control. And then this is looking at challenges of technology. So the biggest one, device malfunction and device maintenance. So that was the biggest one by far. Engagement, like some kids don't want to wear them. A lot of teenagers don't want to wear it. I don't want everyone to see my device and ask me what it is. Education, size of the device, using the phone and just problems with the app or, problems with the phone. And then insurance, which has gotten much better. Medicaid in Ohio also covers CGM extremely well now for our patients. So that's a big victory too. Dr. Wood helped make that possible with her advocacy. And then now looking at the intervention we have planned, because we want to plan a study where the people we're doing the study for think it's gonna be something beneficial and get input and ideas. So this goes to the importance of stakeholder feedback. With Julia is also working on studies with stakeholder feedback. So this is the proposed intervention, the perception of the community health worker. So sharing knowledge, tips, advice, and resources, just someone there who understands diabetes. And our community health workers also work as medical assistants for us. So they do it in and out every day. So they're people who really understand the technologies. They put them on people and troubleshoot things and download devices. So they really know what's going on with the devices. Good for motivation, a check-in that's hands-on, and then a liaison, which is the basic idea of a community health worker. It's not the doctor or the nurse telling you what to do. It's an in-between person helping you to kind of put into action all the things advised by the team. And then this is looking at the perception of the dashboard and app. So quicker and better communication, proactive outreach. We're reaching out to patients and then keeping them informed, good for diabetes management. And then unintended consequences. Of course, things can come up that, you might not think of ahead of time. So just thinking ahead what might happen. Of course, confusion, miscommunication, human error. What if, we don't reach out one week because something happens with our dashboard. It's not flowing right. Again, the app could always malfunction. What if someone misses a notification or alert? We also asked our families, how many times is too many times to call? If I see you in the red zone, how many times can I call you before you're going to get really annoyed at me? So we talked about that. 3 was the magic number. And then, what if patients don't engage? And then privacy and data, because all this data about the person is, flowing from the continuous glucose monitor or the pump into this app, into our dashboard. So a lot of data connection points. But overall, the feedback was really positive. And so we're going to get started with this in a month or two. So this is kind of showing what the dashboard will look like. So these are 2 people who are in the red zone here. And it could be for, again, too much hypoglycemia, low time in range, not wearing the device. And then this is kind of showing, another way to look at it. This is the app on the patient side. I don't know if you can read this very well, but it says, great job, you are meeting your time and range goal this week. We're going to send positive reinforcement messages weekly, because that was something we got in the feedback. I don't want it to all be, if I'm not doing good, I don't want it all to be these pessimistic messages. So we added a positive reinforcement message every week, no matter what. And so we're hoping to get 60 participants. It's going to be a completely African-American Black population, because that's who this is designed for. And we're excited to get started. And this is the team working on this with me. So thanks.
Can I ask a question? Are your community health workers members of the community?
They're both Black. Is that what you mean?
Yes. Mm-hmm.
Yes.
Thank you so much, Dr. McLeish. One thing that JDRF is working on right now at the intersection of research and regulatory, that will be will be feeding into clinical trial design is some of these patient inputs or patient outputs. So what I mean by that is a drug, intervention, treatment. What are the benefits that patients are reporting from them? They call those patient-reported outcomes. So this is one of the things that Dr. McLeish clearly and their team clearly engaged their patient community in what they thought was important in terms of the benefit and the outcomes they were hoping to achieve. And so we're looking to standardize these things because it makes clinical trials much more efficient to have a standard set of outcome measures. So now our final speaker is Dr. Amjad Ali. Some of you may have seen him earlier if you had a chance to visit a lab, and he's going to talk a little bit about his work as a third-year fellow moving from bench to bedside.
Thank you. Thank you for having me. Thank you, JDRF and the organizers, for giving me a chance to speak at this platform. Today, I'm not going to take more of your time explaining about any particular research. I want to take some time explain to you guys regarding different aspects of research, including bench research up to clinical research, and try to build a bridge between them and trying to understand the different aspects and different processes that develop from an idea all the way to a medication. So first of all, let's acknowledge the profound impact of type 1 diabetes that has on millions of individuals worldwide. They say that there's around 8.4 million people with type 1 diabetes currently, with it expected to increase up to 17 million in 2040. So unraveling the mysteries of type 1 diabetes and developing new therapeutic options requires a close collaboration between different aspects of healthcare, including the scientists, clinical researchers, and everyone in between, with the final goal of improving the overall day-to-day life of people who are living with type 1 diabetes. So for that, let's go on a journey. Let's start to understand how an idea can manifest into an actual medication. So starting with in order to have an idea, we need to understand the actual processes that develop in a patient who have diabetes or any disease in general. And that's where bench research comes into play. Bench research, by the term, means any kind of research that is conducted in a laboratory setting. So any kind of like what we call in vivo, which means inside the organism, or in vitro, outside the organism, but it is usually done in the lab setting in a controlled environment. The term bench comes from the laboratory bench on which all the lab research is done. Bench research aims to understand the disease processes on a fundamental level. By the fundamental level, I mean on a cellular and molecular level. And why this is important is it is only by understanding these processes that we are able to understand what kind of the different targets that we are able to have for therapeutics. So in terms of diabetes, so in bench research in type 1 diabetes, as Dr. Ramakrishnan and others have all been involved with, we try to understand diabetes on this specific cellular and molecular level. This includes study of genetic predispositions, the study of immune dysregulation that happens with people who have type 1 diabetes, as well as the beta cell dysfunction that overall leads to people having type 1 diabetes. And how this is first we try to understand why this happens. So the question why is this happening? So it is only by understanding the why we are able to understand the different mechanisms that lead to a person having diabetes. In understanding this, we are able to find out different therapeutic options, different targets that we can use with the goal of trying to reverse type 1 diabetes. In terms of the current options that we have with diabetes, including the management and the development of the synthetic insulin, development of stem cell research in terms of regeneration of the beta cells, in terms of immune regulation in order to prevent diabetes, the study of gut microbiome in type 1 diabetes, all have a final goal of trying to translate research from a cellular level up to the individual level. So this bench research actually serves as a starting point, starting from the cells all the way to a patient. So for example, like Param has been doing in his lab, we try to understand a specific process on a cellular level that leads to diabetes. Now we think we have a hypothesis, which means we kind of test our hypothesis. Does changing a protein or changing a specific mechanism, will that change or will that reverse diabetes? So this is something that we test on a very basic, on a very molecular level. And if we are able to see that is then need to be translated to patient population. So the goal of bench research is a starting point with the final goal of reaching the target to the patients. Okay, so imagine, so I did my research. I have something, I see like modification of this protein leads to decrease in the incidence or decrease in the severity of type 1 diabetes. Now, what do I do with this information? So that is the next step, what we call like translational research, which means trying to replicate the promises and the discoveries of bench research onto an actual individual level. So this is a very exciting phase of research where we actually try to see, are we actually seeing the same differences? Are we actually seeing the same results that we are seeing on a cellular level, on an actual individual level? And it is understanding that our body is very complex. There are, along with, for example, if I'm studying a particular process on a cell, we have to understand there are multiple other processes happening in our body which can co-interact and which can actually kind of give another picture compared to what we're seeing in the cell level compared to an individual level. But this is a very exciting phase of research, because if we are actually able to see any kind of improvement or any results, that means this is the next step to actually get developing a therapeutic object. Okay, so imagine, now I'm in my research. I got this protein modification, and it seems to be decreasing the incidence, or it seems to be decreasing diabetes in a particular individual. What do I do with this information? Now so that is the next step where the clinical trials come into place. So clinical trials are a way of trying to get these discoveries and making it into an actual therapeutic agent. So with well-established clinical trial, these clinical trials are a proving ground for our bench research to actually see, does it actually work in an individual? We established that it's working in a cell level. We established that it's working on an individual level. Now, does it work on an overall population level? That is the mainstay of clinical trial research. Clinical trial research, always the first most important thing before we test the efficacy of any medication is to make sure they are safe. So the number one intention is to make sure it is safe to use in patients. And then the second step is to actually see on an overall large population, does it actually work, and are we seeing the effects that we like to see? All the research, all these clinical tools that we use on a daily basis, the checking of glucose, the hemoglobin A1C, checking antibodies, that on a daily basis that we do in the clinic, checking for urine ketones, it is important to know that all these have been derived from bench research with long-term use and successful translation of data from cellular level all the way to the patient level. And that goes the same thing for therapeutic options. As the advent of insulin, as we continue to develop more synthetic insulin and different forms of insulin, the different antibodies that we use for detection of diabetes on a daily basis, the new medications such as teplizumab that has been showing promising results to actually delay the progression of type 1 diabetes, as well as insulin pumps. All these have come up from a very close collaboration with scientists, clinical researchers, and everyone in between, and which is a very dynamic and dynamic process. So that's how it is done. So you have, in order to get a medicine, first understand what's going on a cellular level, then trying to see what are the areas that we can actually target, that we can change according to what we need, then trying to replicate on an individual level, and finally on an overall population level. If you are able to successfully do this, then you have your next medication. It is not to say that this process is not without any challenges. So you can actually think of millions of challenges that you might encounter in this process, starting from a bench research, actually getting this reproducible data and trying to get the same data all the time is another challenge. Second thing is actually trying to get the same data on an individual, on a human being level is another huge obstacle we all face in research. Once again, knowing that our body is very complex. We don't know how our body is going to react to anything. We ourselves don't know our body. So having something new being introduced to our body, it's always a challenge. Will we see the same results on actual human body? Then always it comes to the question of ethics, such as trying to make sure this is actually safe for the patients, actually improves the patient, and it improves the overall life of the patient, and not just focusing on one particular aspect that we are trying to improve. So definitely there are some challenges that everyone encounters in research, but overcoming these challenges and getting new results and seeing the product of our research in real life is what drives all of us researchers towards continuing research in our careers. Going next to the future objectives. So in terms of type 1 diabetes, there has been a lot of promising researchers like our panelists have discussed here. In terms of autoimmunity, we continue to understand the very complex and intricate mechanisms that our body has in terms of our immune body, the different steps that's involved in our immune system, and trying to see in what protein or what step can we kind of alter or modify that can help us to at least slow down or reverse this autoimmunity that happens in type 1 diabetes. And with the current technological data, artificial intelligence, and data gathering, this also has really helped us to advance our research at a very rapid level. I'd like to conclude that research is a very dynamic process which requires a very close collaboration between all aspects of healthcare, including the patient, who is like the most valuable person we have in our research. As we continue to make more stride and try make more understanding of the disease processes, I believe we are one step closer to hopefully developing a cure for diabetes somewhere in the future. Thank you. Once again, my objective today was to just kind of explain the process and explain the cycle that happens with diabetes and in research, because I know sometimes it can be confusing when we hear about different aspects of research and how it ties all together into actually benefiting the patients. So I hope this helps, but thank you very much.
Long page, and I would be remiss if I didn't invite our next speaker, Dr. Apollonia. Sorry. Oh no, I should not do that. Miss Apollonia is a third-year MD PhD student, and she's going to talk to us about using RNA to potentially provide therapeutics in type 1 diabetes. So I apologize for my mistake. I was on the wrong page of my paper.
No worries. I want to thank everyone for coming today. So like was mentioned, I'm an MD PhD student currently, meaning that when I graduate in a few years from now, I hope to become a research scientist, like many of the speakers today, with a current interest in pediatric endocrinology. So I'm going to talk today about RNA-based therapeutics for type 1 diabetes. And like Amjad was just saying, this is kind of very much the bench side of the research cycle that happens. And so our goal of my research, and also the lab as a whole, is to identify new signaling pathways with the ultimate goal of using the information from these signaling pathways to develop new therapies for type 1 diabetes. And so I'm going to introduce some important cell types that'll be important for my presentation. So first we have a CD8 T cell. I like to think of this T cell as the protector of the immune system. For example, they will kill cells that are infected with viruses. So this function is represented by the bow and arrow. The next cell is the CD4 T cell. I like to think of these cells as the orchestrator or the conductor of the orchestra. And that's because these cells produce a lot of factors that help other cells. So they're just producing factors that will help other cells grow and function. The third cell is called a Treg cell or T regulatory cell. I like to think of these cells as the traffic guards of the immune system. So similar how a traffic guard will stop the flow of traffic if needed, these cells will stop other cells from growing if it believes that these cells are harmful in any way. And then the fourth cell is the beta cell, and these cells are in the pancreas that produce insulin. So these functions are present in individuals without type 1 diabetes. And patients with type 1 diabetes, these cells are kind of evil, right? So you have their CD8 T cell and your CD4 T cell. These are evil based on their facial expressions. And that's because they're self-reactive. So the CD8 T cell, instead of killing cells that are infected with viruses, they're killing now your own pancreatic beta cells. And your CD4 T cells are producing factors that aid the CD8 T cells in killing the beta cells. And then your T regulatory cell is just sad. They're usually depleted or they're not functional, so they're not able to stop traffic. And these cells are just going to keep proliferating, leading to more destruction of your beta cells. And like I said, your beta cells are destroyed. So going back to RNA-based therapeutics, this is really a growing field. There are currently 18 clinically approved RNA-based therapies. The one that's obviously very known right now is the COVID vaccine, which is an mRNA-based therapy. And so mRNAs in the body are translated to protein. And I'm specifically focused on microRNAs. So microRNAs are small RNA molecules that bind to RNA and prevent protein production. So they're specific for a specific RNA sequence, and it stops that RNA from getting translated into protein. The specific, so microRNAs are expressed in a wide variety of cells in the body, including immune cells, such as our T cells. And they really play a role in just keeping the immune system kind of in that balance of good and evil. And specifically, I'm focused on one microRNA that we call microRNA D. And I'm focused on this microRNA because it's been shown in patients with type 1 diabetes that there's an increased levels of this microRNA, which leads us to believe that it could be playing a role in disease progression or disease onset. So my central research question is what is the role of microRNA D in the development of type 1 diabetes? So to study this, some of you saw this earlier, we use mouse models. We specifically use a NOD mouse model, which is a non-obese diabetic mouse model. And these are really helpful because these mice at a certain age spontaneously develop a phenotype that's very similar to type 1 diabetes. So it more closely mimics human disease compared to other mouse models. And so about 12 to 15 weeks of age, these mice will become sick, which is represented in that lower picture. Usually it manifests in the eye, and they have like a scrunched back. And we can measure their blood glucose to confirm that they actually have developed type 1 diabetes. And how we use these mouse models is we create genetic deletion models, so we can delete proteins, or in this case, we deleted that microRNA D from these mice, and we can compare the disease manifestation in wild type versus microRNA D knockout mice. We measure diabetes developed using blood glucose monitors, very similar to what happens in humans. We start at 8 weeks of age and go to 35 weeks of age. We also collect the pancreas and the pancreatic lymph node from these mice. So the pancreatic lymph node is shown here. It's just attached to the pancreas. And this is kind of the home of these T cells that I was talking about earlier. So that pancreatic lymph node is very small. It's just a concentrated small ball of immune cells, such as T cells. And so this is our initial data side. So we've shown that when you knock out this microRNA D from the NOD mice, they do not spontaneously develop diabetes. At least they do, but to a much lower degree than the NOD wild-type mice. So in black, you have the NOD wild-type mice. So you can see by about 12 weeks of age, 90% of the mice spontaneously develop diabetes. At contrast, at that same time point, only about 15% of our knockout mice have developed diabetes. So to put that into kind of a schematic, our wild-type mice more closely represent this sick mouse with the high blood sugar readings, and our knockout mouse more closely represents this healthy mouse with normal blood glucose readings. When we look at the pancreas, you can see that when we knock out the mirror D in these mice, they do not develop encephalitis, which is the presence of these CD4 and CD8 T cells in the pancreas. So again, in our wild-type NOD mouse, you can see the encephalitis is right around here, which is just the collection of T cells in the islet in the pancreas. In our knockout mouse, there's no such infiltration of these CD4 and CD8 T cells, and that's just schematically represented in this bar graph, with the black being the wild type and the blue being the knockout. So again, schematically, we can show that in our wild type NOD mice, there's tons of these angry T cells in the pancreas. Comparatively, in our knockout mouse, they're still there, but they're to a much lesser degree, indicating that these harmful CD8 and CD4 T cells aren't in the pancreas and aren't able to destroy the beta cells, which is likely why they're not developing diabetes. So the final data slide is talking about the T regulatory cells. So remember, these are the traffic guards of the immune system. And in that pancreatic lymph node, we have shown that the knockout mice have increased T regulatory cells in the pancreatic lymph node. So these cells are able to function to stop the proliferation of these autoreactive CD8 and CD4 T cells. And this is able to happen to a greater degree in our knockout mice. So again, schematically, your wild-type mouse pancreatic lymph node is going to have, they're going to have these T regulatory traffic guards. But in our knockout mouse, the T regulatory traffic guards are increased.
Are they smiling too?
Yeah, they're smiling. There's probably some sad ones, but we're trying to be positive. So overall, what I've shown here is that when we delete microRNA D from our NOD mice, they have increased traffic guard Treg cells in the pancreatic lymph node, and these cells are then able to inhibit the proliferation of our evil autoreactive CD8 and CD4 T cells, and therefore we don't get destruction of our beta cells in the pancreas. Therefore, these mice that have the knockout of microRNA D are not developing spontaneous diabetes. So how we can use this as a potential therapeutic option, they make anti-microRNAs, and these are really common. There's several already in clinical trials. So this is a schematic I showed earlier where a microRNA is binding to mRNA, stopping protein production. You can add an anti-microRNA into the system. This is specific for, our microRNA D. It binds to it based on the sequences, and this essentially mimics our knockout mouse because this microRNA D is no longer able to bind to its mRNA sequence. And so this is a potential therapeutic option that can be used in humans. We have to do initial studies in mouse cells and mice first, obviously, but this is how this bench research can potentially be translated into a therapeutic option in the future for type 1 diabetes. I want to thank everyone for listening today and thank my lab and my funding.
Thank you so much. That was a really fun way to talk about autoimmunity. I don't think I've ever seen such a fun way of doing it, and that was very entertaining. So thank you. But I'm absolutely thrilled to have you all here tonight. Before we start Q&A, because I know some of you are probably going to have to take off fairly soon, and if you need to, that's okay. So first, I just want to thank all of our panelists. Very grateful to all of you for sharing your passion and enthusiasm for changing the lives of people with type 1 diabetes, whether it's from the bench or at the bedside through clinical access. I think these folks represent the entire spectrum of research, and JDRF is certainly involved in all aspects of research, although not everybody on the stage is a JDRF-funded researcher, but all aspects of research that their presentations illustrated. And I I set out at the beginning that it was our hope that tonight would inspire you. And so I just want to say, please be engaged. We need your help as volunteers, as advocates, as donors, as fundraisers. The success of work like the research work that was presented tonight relies on our community to advance. So I hope that you are inspired, and I'm really grateful to all of you for being here. And please stay engaged with our community that none of us wanted to join, but that is the best one ever. So with that, we do have opportunity for Q&A if anybody has any questions. Let's see. How's the best way to do this? Let me come down. I'll come down. There we go. That was graceful.
You told me not to do that.
Oh, I told you not to do that. Okay.
I could have talked a lot. So this is for the last speaker, Miss Apollonia. I like your name. I was actually really interested in your research on a basic science level. The microRNA D, what is it binding to? It's an endogenous signaling molecule. What's its normal function? What's it binding to? Is it like a specific sequence or is it a general term?
Yeah, so it's a specific microRNA. We know the sequence it binds to, and it's very endogenous. It's a normal microRNA that everyone has in your body. It's specifically involved in a pathway called the NF-kappa B pathway, which is involved in many things in the body, but generally inflammation and immune homeostasis. And so we have shown that this microRNA is functioning as expected in our system. We can measure this by looking, like I said, so when this microRNA binds to certain mRNAs, it leads to decreased protein expression. And so the two, we know the mRNAs it's binding to, and we've shown in the lab that it is leading to expected decreases in protein expression. And so that's kind of the general pathway it's working on, is the NF-kappa B pathway. And we haven't shown yet in our system exactly how it's completely functioning to reduce incidence of diabetes. And that's the work I'm going to be continuing on throughout my PhD.
Thank you. Other questions? Oh.
You can say that. I can talk about it.
Oh, now I'm coming.
I got a question because that's really cool stuff.
Hold on. They're recording this.
Okay.
Thanks,
I
Ted.
might change my question. Dr. McLeish, really cool study. I, as a parent of a college age type 1, I'm wondering if there's any opportunity to clone the system, enroll college students that are with type 1 to have the, rainbow guardian floating over them and keeping them a little more compliant. Maybe that's an idea on how to spin it out for, new income stream.
And to clone that system for not just college students who might be rainbow patients, but for college students who might be patients elsewhere. Having 2 college students who are type 1 right now.
So yeah, we hope this is just the beginning. We're working with this company called Keyboard Med that built everything for us, because I could not build an app or a computer program at all. So this is like the first time we're trying it out, but we hope we'll get to use it in more situations in the future. And the dream would be we use it for all our patients, all 1,000, rainbow diabetes patients, and then we can flag whoever we need to reach out to who's not doing well or that needs help.
Well, and I might just add, one of JDRF's most frequent research partners is the Helmsley Charitable Trust. I'm sure most of you are familiar with them. And one of their research priorities is to reduce disparities in health outcomes along with the T1D exchange. So I'm sure depending on the outcomes of your research, there might be other people interested in how it goes and in expanding on your work. So best wishes for you as you proceed. Other questions? Oh, sorry. Let me do this one here first, and then I'll come back. Hey, I'm getting in a car for 3 hours, so this is good.
Thank you. I'm just curious to hear about how often, like in the clinical setting, like immediate siblings of type 1s are being tested for the antibodies, and like how often like teplizumab is then used in practice based on that, and like insurance coverage of that.
So I'm going to just make a couple of comments before our panelists answer. So one of JDRF's research priorities is screening, and our ultimate objective would be global universal screening. Standards of care that are put out by our partner organization, the American Diabetes Association, do now recommend that first-degree relatives of people with type 1 diabetes be screened. I ran a one-person research project this spring and took my son, who has been screened in a clinical research environment before, to his pediatrician and asked for him to be screened. I was fortunate enough to have worked with some of my JDRF colleagues to know exactly what lab tests to ask for, because when I walked in, our pediatrician said, absolutely, we'll be happy to screen. I don't know anything about these tests, and I don't know anything about the results. And I said, that's okay, I do. So implementation is a challenge. And the other big challenge about screening right now, even though it's recommended, is that there are not consensus guidelines yet on what to do if somebody is antibody positive. So what JDRF has done is brought together working groups of scientists, clinicians, et cetera, from all over the world to address monitoring guidelines. And we should be publishing a paper pretty soon. Okay, now you go ahead, then.
Great question. There are, so ADA as well, I'm in the leadership of ISPAD, which is a different society for pediatric and adolescent diabetes. We also have a similar guideline recommending screening. And there's different ways to go about it. And so you can be screened through a research trial like TrialNet. You can have a clinician order the different antibodies. There's also antibody screening kits that you can get. There's a couple programs out there, T1D Detect, and
So just 1 second, all the kits are on hold right now through the FDA.
for
All home screening kits are on hold right now through the FDA. So,
but the
but they'll
lab,
come back.
yeah, and so I'm part of that JDRF working group that she talked about in terms of once you know somebody's antibody positive, how do you then monitor them through, maybe not even progressing to multiple antibodies or to stage 1, 2, eventually stage 3 diabetes. And so that guidance is coming out soon. ISPAD is one of the endorsers of it too. So I saw a preview of it a couple of weeks ago. But then with teplizumab now FDA approved, and we were at our ISPED meeting a couple of weeks ago, and the PROTECT study, what the results released there showing also benefit for people with new onset stage 3 diabetes, showing a similar benefit. So we're here at Rainbow. We are going through the steps of being able to offer Tzield to patients. We have it now on formulary in our pharmacy. We have our clinical protocol written, and it's going through committees for approval. So we should be able to offer Tzield very soon to patients that are antibody positive in stage 2, and maybe the FDA eventually will also approve for stage 3 based on the PROTECT results.
Yeah, I mean, it's our hope at JDRF that Sanofi, which owns the drug, will go back for a label expansion to stage 3. And we're certainly hearing about clinical implementation barriers. And I know Sanofi is very much invested in that. But what they've asked JDRF to continue to support is agnostic to treatment, because we hope that Tzield is just the first of many such treatments. Things like more screening, more clinical trial participation, and to support clinical implementation or adoption to the best that we can, knowing that we're not an organization like ADA that has all the providers in our network, but we are partnering with ISPAD and many, many others. It's crucial that this drug be successful because we want more drugs to be successful. So we're all coming together. I think that's the takeaway. We're all coming together to make sure that we can promote its success. And I'm so sorry, I didn't realize you were on one of the working groups, or I wouldn't have stepped in. But thank you so much for being in the working group, because it's really, really important work. I mean, it's basically unethical to have somebody go out there, get monitored or get tested for antibodies, and then like the burden of knowing that without any kind of standard of care after that is unacceptable.
There's a whole working group too that's focusing on the support and that a family needs to go through this process with another child or themselves. And so we have a whole group of psychologists that are working on how do you support a family through testing for antibodies and then progression. We also have an entire working group that is focused on educating primary care providers of how to screen and what to order, and then at what point do they need to come to endocrinologists. And so it's a pretty comprehensive.
Yeah, it's an amazing, amazing effort. So thank you for being a part of that. I know Kathy had a question. I'm grabbing the closer ones first.
I need to put on my reading glasses. I'm sorry. For Dr. Ramakrishnan, you in your bio, you have that your findings suggest that gestational diabetes may predispose the fetus to autoimmune diabetes. Could you speak a little bit to that?
That's a great question. Because we talked about a sugar modification, a protein seeing a little bit of sugar becoming hyperactive, and then it is supporting the bad T cells and killing it. So just to note, the women with hyperglycemia, the last trimester of pregnancy, they have high blood sugar. And the only compound that passes the placental barrier to the fetus is nothing but glucose. The placenta can protect the fetus from most of the environment, but glucose is free to pass in and out. That means the fetus inside the placenta is exposed to hyperglycemia during the last trimester of pregnancy. That means the T cells that are developed within the fetus, they are already exposed to high sugar and they are becoming hyperactive within the womb. So I showed you one picture. The youngest patient recorded with type 1 diabetes is 10 days old. 10 days is not enough time to get type 1 diabetes. That means the fetus is already predisposed to the glucose in the womb, and they are having hyperreactive T cells and destruction of beta cells, so that they are born without enough insulin to support them. So that is a key link between gestational diabetes and type 1 diabetes. So the glucose that passes from the mother to the fetus can activate T cells in a bad way so that it kills the insulin producing cells in the body. So that's as early as the treatment starts, it should work.
So I just want to make sure that I'm not aware of any clinical data. I don't know if my other colleagues are aware. Just sometimes what you can see in animal studies and implementation of pathophysiology might not be necessarily yet observed in human studies. As a clinician who treats humans, so so I just want to make sure. Yeah, so, but it's, I just want to make sure there is an understanding. And also, sometimes when some babies are diagnosed, and my colleagues can talk better, when it's a 10 days old, that might mean there is a genetic issue that is perhaps even affecting the secretion of insulin that we have been seeing more and more clinically, which is fascinating. As I diagnosed someone age 33 during screening for pancreas transplant. That was fascinating. So, yeah, I just needed to say something. No one is asking me questions, so I had to take the microphone away.
Oh, just to continue on that point, I totally agree with that point. So the reason is that not enough studies has been done specifically on type 1 diabetes, but there are enough studies on obesity. A mother with hyperglycemia having a bigger baby or within the overweight baby is very common. That is also same principle of glucose. Possible. Yes. Yes, that's true.
Yeah, and just I was asked to explain the monogenic forms of diabetes. So there are some types of diabetes that are not type 1, that are not type 2. They're called monogenic diabetes, and they're as a result of a genetic change that results usually in insulin secretion. And so we take care of, we have several in our practice that we take care of, and by definition, they're usually diagnosed under 9 months of age, and we still measure antibodies, and they're antibody negative, and we can send genetics and usually find out there's multiple different types, but it's a form of non-autoimmune diabetes that's diagnosed in the very young.
And it's treated the same as type 1.
Most types are treated the same. There are a couple types that are responsive to oral sulfonylurea medications that so that they can be successfully treated without insulin, but only a couple of them.
Thank you. Appreciate it. Thanks also for making your point about animal models versus how it sometimes can be in humans. Okay.
Yeah, this is for the whole panel. So you guys look at statistics all the time. A lot of you guys talked about statistics, but and Dr. P, I think you even mentioned in yours the statistics in the U.S., right, for type 1. How's that compare across the globe? Do we have any studies that show statistically which country is the lowest, which country is the highest, and is there any thought behind that?
So before they answer, I just wanted to alert you to a tool that JDRF Australia and JDRF International partnered with a number of organizations to develop. It's called the Global T1D Index. If you just Google it, Global T1D Index, for any country in the world, we have a validated algorithm. It was published in Lancet, the Lancet Diabetes, which is a highly respected journal, that will predict the number of the incidence, the prevalence, the number of lost, and the number of missing days due to type 1 diabetes. So it's they're working on version 2.0 right now, but it's a pretty accurate picture of global type 1 diabetes. And what it shows you very interesting is what one small change, for instance, we're doing a small pilot in sub-Saharan Africa with our partners at Life for a Child and CHP, I can't. Anyway, where you put up posters about the signs and symptoms of type 1 diabetes that cost maybe, less than 10 grand to do, and you can increase the rate of diagnosis significantly and prevent people from dying without being diagnosed. So it's a really interesting model. But to answer your question, I would ask the panel to comment.
There's a study called SEARCH, and it's Search for Diabetes in Youth. And that has given us the best idea about the global rates of type 1 and type 2 diabetes in children. But in type 1 diabetes, they do it by country, they do it by race and ethnicity, ancestral background. But the take-home message is that globally, the rate of type 1 diabetes is increasing 3 to 5% or greater every year, and that is greater than what we would predict based on just genetic alone factors. So this is what's really driven the idea that there's something in our environment that is triggering the autoimmune condition and causing these rates to just continue to accelerate globally. They're not plateauing. They continue to accelerate. That's why some have shown that, by 2050, 2060, the cases are going to go up. And it's also shown us that the highest incidence for new cases is under the age of 6 years. So there's another big study that will be publishing results soon. We've seen some of it, called the TEDdy study. And this started at the Barbara Davis Center in Colorado and some other sites where they've literally been following children from birth, infants from birth. What formula were they fed? What infections did they have? When did they have their immunizations? Everything from birth. Their whole medical history to try to understand the multiple environmental factors that might be triggering this. And so TEDdy is reaching its 15-year, I believe. I just heard a symposium by one of the co-PIs, principal investigators at the EAS, the European meeting. He won a national award, but this is where we really understand, what happens when you have one antibody, what happens with two antibodies, how quickly you're going to progress to type 1 diabetes or not, and even like which antibody presents first and then second. All of this matters. And so it's this huge database that I think is going to really help all of us, the community, JDRF, ADA, all of the investigators understand what's our next target, where do we go, and how do we prevent this acceleration in the rate of type 1 diabetes. I don't know if anybody else had it.
I want to make two comments, if you don't mind. First, in addition to TEDdy, what JDRF did is took these data sets that's from the US, that's an NIH-funded study. They took these data sets from a number of countries and used IBM Watson machine learning and AI to bring them together. And we've been getting a lot of great data out of that. Things like what are the optimal ages to screen, more information on the order of presentation of autoantibodies and how it contributes to progression. So that is some of the work that we did. We kind of were a convener of multiple data sets. There's a big data set in Finland. There's a big data set in Bavaria. And so we've been working to pool all of this data and use it for, deducing a little bit more information to your point. One other thing I will mention is none of this is to speak, all these data sets, they're on kids. And none of this at all comments on the fact that many, many people are being diagnosed now as adults, and that 80% of people living with T1D are adults, and that T1D in adults looks really, really different in its progression than T1D in kids. And there's a huge amount of misdiagnosis. So another project that JDRF has funded is working with IQVIA, that has all these clinical trial records, or all these EHR records, electronic health records available to them, to maybe make an algorithm that would point clinicians to screening for type 1 in adults versus going to the default that sometimes happens, not in endocrinologists, but amongst primary care providers. Quite frequently, they just say, oh, it's type 2. And that misdiagnosis can be really detrimental. So a lot of work going on there.
I have seen people go to the emergency rooms and then seeing physicians and getting metformin.
Mm-hmm. Yeah.
I have seen it. Yeah, it is very unfortunate, but it's there.
Any other comments on the increased incidence and prevalence of T1D? Okay. All right. I know Kathy had a question, and then I think we'll wrap it up because I'm sure most of you have had a long World Diabetes Day now that I think it's 8 o'clock. And I know that a number of us are going to drive back to that other state north of here tonight.
Just very fast. Don't go far, Elizabeth, because I think this might be for you too. But first, I want to say thank you to the brilliance we have on the stage, and thank you to you too, Elizabeth, for sharing your insight on JDRF. But it strikes me being here with you all tonight. I'm so grateful for the passion you have for your field and grateful on behalf of all of our T1D families. But I remember starting at JDRF about 13 years ago, and we had 5 human clinical trials. How many do we have now?
So JDRF is funding between 70 and 80 human clinical trials right now. That's not to mention industry-sponsored trials that are related to research that came out of our bench or discovery science work. And it's not to mention, I mean, a lot of the trials of the clinical work that was described tonight here by our panelists isn't necessarily funded by JDRF. NIH is a huge funder of clinical trials, so I cannot recommend enough, and I'm sure you guys might want to put a plug in too. Go take a look at the Clinical Trial Connections tool. I mean, some of these clinical trials are as simple as filling out a survey. And you really can make a difference. It's very, very crucial. Science won't advance without it. So you guys might want to put a pitch in too. Go ahead. Put a pitch in for clinical trials.
Absolutely. And I think you mentioned earlier, TrialNet is an NIH multi-center international consortium really targeting autoimmune and trying to have early intervention. So I sit on the National Data Safety Monitoring Board for TrialNet. So I can't recruit you in the TrialNet, but I can tell you that, you know, this is where a lot of the new discoveries, along with JDRF, this is where it's happening. Their goal is, now we have teplizumab. What's the next one? We can delay it, but we can delay it. Then we need to have the second drug to give people, and the third. I think it's always been thought that this will probably be a cocktail because of the diversity in the human condition, which you've heard. But they're on their way, and so we're currently monitoring 4 or 5 TrialNet studies, all really new targets. We talked about RNA, microRNA, really novel new approaches that will add on top of each other. So there'll be additive therapies on top of teplizumab, different targets, different approach. That's how we're going to, I think, cure type 1 diabetes.
And I'll also add that similarly in Europe, there's a clinical trial organization called Anodia that JDRF is also a huge partner of, a huge funder of, that's doing the same kind of thing. These disease-modifying therapies in combination, and those are taking place in Europe, the UK, and Australia. So I think the cool message I can take away from all this is that everybody is talking, collaborating, convening, coming together around, to your point, what's the next thing, what's the next step, whether it's at the bench or at the clinical trial level. And I really hope that you can take that away from today. Any other? So thank you again to our panelists. You guys were amazing. I will really appreciate everybody kind of rolling with the punches tonight when we had a little bit of schedule confusion. Grateful to all of you for participating, and thank you to our T1D family for being here. I hope that all of you feel super inspired after all this, and we really need your engagement. So thank you so much.